Good morning, everybody. Welcome to a Stem Cell Technologies educational webinar. My name is Dr. Javier Menendez. I'm a research scientist in our antibody development group here at Stem Cell Technologies. There will be two parts in today's conference, a 30-minute presentation segment and a 10-minute Q&A session. The Q&A session period will be limited to 15 minutes. Any additional questions outside of this period may be addressed to Stem Cells Technology via email or telephone. To ask a question during this presentation, please use the question box located on the right of the slide. In today's webinar, Dr. Jenna Capyk, product specialist at Stem Cells Technologies, will talk about the probability of monoclonality for limiting dilution cloning compared with semi-solid cloning. Let me now hand you over Dr. Capyk.
Hi, thanks Javier. So the purpose of this webinar today is to explore two different methods of single-cell cloning that can be used during cell line development. So we're going to look at limiting dilution cloning and semi-solid cloning. So I'll be talking a little bit about the mechanisms behind these two cloning methods, as well as how to estimate the probability of monoclonality for cultures that are isolating using both of these techniques. So for limiting dilution cloning, I'll be exploring why the probability of monoclonality is often overestimated for cultures that you've isolated using limiting dilution. And for semi-solid cloning, I hope to illustrate that this method makes it very easy to isolate cultures that have a very high probability of monoclonality. And this is for each round of cloning, meaning that because you have a high probability of monoclonality for each round, you require fewer rounds of cloning using semi-solid cloning versus limiting dilution, ultimately leading to shorter cell line development timelines. So first, let's ask the question that sort of underpins everything. Why do we want to have monoclonal cell lines? Now, of course, many of you are probably very familiar with this, but of course, the end result, the desired result of many cell line development projects is the production of a very specific protein product. Now, of course, these molecules are large and complex, so instead of using chemical synthesis methods, often mammalian cell lines are used to produce these products. And the cell lines that are used are monoclonal. So again, as many of you know, there are many reasons why monoclonal cell lines, rather than mixed cell populations, are used to produce these proteins. First off, using a monoclonal cell line goes a long way toward having consistent production of the product that you want throughout the cell line development and production process. And this consistent production is required in order to carry out the detailed characterization that you need. Using a monoclonal cell line also makes it easier to purify the substance, as the background of interfering substances is going to be lower when you're using a monoclonal cell line. And you have a greater culture stability, meaning that you have the ability to do multiple production runs using a monoclonal cell line. And this is something that absolutely can't be said for a mixed cell population. So all of these are important considerations for safety, economic, and regulatory reasons in biomanufacturing. So if we take a look at a cell line development process, the first step is to either introduce your gene of interest through transfection or to conduct a fusion of splenocytes and myeloma cells to produce hybridomas. And this generates a cell pool that you're then going to use to generate your cell line. The next step is to take that pool of mixed cells and separate them out and clone them out into individual monoclonal cultures. These cultures are then screened to identify those that are producing the product that you're looking for, and then those most valuable, interesting cultures are cultured under conditions in which they're producing a high amount of your desired product. In this presentation, I'll be focusing on this step, the cloning stage of this procedure. And this is a critical step, as cloning of some kind is absolutely required in order to generate a monoclonal culture. So as I mentioned, I'll be talking about two different cloning methods, limiting dilution cloning and semi-solid cloning. And I'll be going into some of the basic mechanisms of how these two methods establish monoclonal cultures. And through looking at these basic underpinning mechanisms, I hope to illustrate that for semi-solid cloning, this method makes it very easy to isolate cultures with a very high probability of monoclonality, again leading to fewer cloning rounds that are required to isolate the cultures that you need, and ultimately leading to faster cell line development. So let's first take a look at limiting dilution cloning. Here we have a mixed population of cells diluted in liquid medium. And then this mixed population is distributed over 96 wells. So, well, 96 well plates. So the basic mechanism by which limiting dilution cloning hopes to achieve monoclonal cultures is by isolating individual cells into unique wells of the 96 well plates. And this is accomplished most often by distributing the cells such that there's an average of less than 1 cell per well. So diluting to this degree increases the chances of being able to isolate individual cells into unique wells on the plate, but it also, as you can see on this plate, results in a lot of empty wells. And these empty wells are consuming project resources. And this can be in the form of cell culture media that's needed, obviously, to be in these wells, as well as the physical space and resources needed to manage all those plates that are required for extra empty wells, and the personnel time required to set up and then process all those empty wells. The cells are distributed randomly into the wells. And this means that while there's lots of empty wells, and you can see that there are some wells that are seeded with 1 cell, no matter how low the seeding density is, statistically, some of the wells will still be seeded with more than 1 cell. So let's contrast this with the mechanism of semi-solid cloning. In this method, the cells aren't suspended in liquid media, but rather suspended as a single cell suspension in semi-solid medium. So this medium isn't sort of rubbery or solid like agar, but more the consistency of a molasses or honey. So it has a very high viscosity, this semi-solid medium, and as the cells are suspended in the three-dimensional growth environment of this semi-solid medium, as the cells proliferate, their diffusion is restricted such that the newly formed cells remain closely bunched. And this means that each of these individual cells is able to grow into a discrete colony in the semi-solid medium. These individual colonies can then be isolated by aspirating them with a pipette and then transferring them to liquid medium in 96 well plates for expansion and screening. So when these cells are seeded in this way, you're seeding each well with thousands of cells from the same clonal colony. And this means that the liquid expansion cultures in the plates have a very high rate of survival, meaning that you end up with pretty much no empty wells. And again, in limiting dilution, you have those resources wasted on the empty wells. So there are a couple of important aspects that really differentiate the semi-solid cloning mechanism. Firstly, again, we have these cells that are suspended in that three-dimensional growth environment of the semi-solid medium, and as they grow, they each grow into discrete colonies, and each colony derived from proliferation of a single original cell. So this means that each of those colonies has a very high probability of monoclonality. The other thing to note is that you can see that you can have large colonies and small colonies, the larger colonies coming from faster growing clones and the smaller colonies deriving from slower growing clones, but all of them form discrete colonies and all of them can be easily isolated. And this makes it really easy to isolate a range of clones with a diverse range of growth characteristics. And this is important because sometimes the slower growing clones can actually be higher producing because of the metabolic trade-off between proliferation rate and productivity. So having a diverse range of clones makes it easier to find those rare high producing clones. And this is in contrast to methods that involve selection in liquid medium. So in this case, the cells are free to diffuse among each other, and as the cultures start to proliferate, they start to be dominated by those fast-growing clones. And this means that you can actually lose the slower-growing clones during the selection process, and that a disproportionately large number of the range of cultures you finally isolate will have originated from those faster-growing clones that might not be high-producing. So let's compare these two cloning methods side by side. So the first step, that transfection and fusion step, is basically identical between the two sides. For limiting dilution, you then have selection and cloning occurring in liquid medium in the 96-well plates. You then screen those cultures to identify the ones that are producing your product of interest, expand the interesting cultures for subcloning, and then each subcloning step is accompanied by a screening step to, again, identify the cultures that you want to carry forward for the next round of subcloning. And the subcloning and expansion and screening is repeated until you have a high enough probability of monoclonality to meet the requirements of your project. So we can contrast this with semi-solid cloning, where after transfection or fusion, however you're going to generate your pool of cells, there's no selection step in liquid medium, but rather the selection and cloning are occurring at the same time in the semi-solid medium, where we're growing those discrete colonies. And then these are picked and transferred for expansion in the 96-well plates. And again, you can see that all of the cultures on this plate are growing. And that's again because each culture is seeded with thousands of cells from the same colony. And like limiting dilution, the next step is then to screen those expansion cultures to identify those that are producing the product you're interested in. But unlike limiting dilution, at this step, after the very first round of cloning, your clones already have a very high probability of monoclonality. So you might still want to do a subcloning step depending on the requirements of your project, but because each round of semi-solid cloning delivers such a high probability of monoclonality, you're going to require fewer rounds of semi-solid cloning versus limiting dilution cloning. So I've been throwing around the term probability of monoclonality a lot, so I just wanted to give a definition as well as a shortened version because that's quite long to write out and say all the time, so you might hear me refer to it as POM. But this is the probability that a culture of cells is made up of genetically identical cells, or that culture arose from proliferation of a single original cell. And I'll be using this sort of as a way to look at how well each cloning method is able to isolate monoclonal cultures. So let's take a look at the probability of monoclonality for limiting dilution cloning. We'll start off by looking at a simple experiment where we have a homogeneous population of cells being distributed over 96-well plates. By homogeneous population of cells, what I mean is I'm not drawing a distinction between those cultures that I'll choose to isolate down the line versus those that aren't producing my product. I'm basically considering all the cells in this experiment as equal. So the cells are distributed randomly, as I've said, and this can be described using a Poisson distribution. So this is a mathematical form that was first described by Simeon Denis Poisson in the early 1800s, and it's the discrete probability distribution that can be used to estimate the number of events that happen over a specific space or time when the average number of events is known. Okay, so for example, you could use a Poisson distribution to estimate the number of apples that are likely to fall during a specific hour of a day if you know how many apples fell during that whole 24-hour period. Alternatively, you could use it to estimate the probability of having a certain number of O's in a spoonful of alphagettis, but only if you took up the time to count up all the O's in the whole bowl and how many spoons you took to eat it. So this is the basic equation for the Poisson distribution that's going to underpin all of the calculations for the probability of monoclonality for limiting dilution cloning. And I'm not going to go too deeply into this equation or to any of the other equations that I'm presenting in this presentation. But if you're interested, there's a video tutorial that is posted on the Stem Cell Technologies website. And this will be sent to you in a link following the presentation. So as I mentioned, limiting dilution cloning is a random distribution that can be described by the Poisson distribution. So the distribution for a plate like the one, or the section of the plate on the left, looks something like this. And so you can see that on the left there's about 70% of the wells that are empty, and that's represented as the y-intercept here, whereas there's about 30 or just under 30% of the wells that contain one cell, and that's represented by this point here. So the possible number of cells per well is shown on the x-axis, and the probability, or the percentage of wells that contain a specific number of cells, are shown on the y-axis. So this equation helps us to estimate the probability that a given well will contain a specific number of cells. So for cloning, we're obviously interested in the wells containing one cell. So this equation can let us estimate what proportion of the wells will contain one cell. But that's not the whole story when we're looking at the probability of monoclonality. For that, what we really want to know is the origin of a culture that we've isolated. So for this culture that I'm interested in, that I have, what is the probability that it originated from a well that was seeded with one cell? And what is the probability that it originated from a well that was seeded with more than one cell? So the probability that the culture arose from proliferation of a single cell, a well that was seeded with only one cell, equals the probability of monoclonality for that culture. The probability of monoclonality can then be thought of as a ratio. So we have the percentage of wells with one cell divided by the percentage of wells that have at least one cell. So this can be thought of as all of the scenarios that give you a monoclonal culture divided by all of the scenarios that will give you a viable culture. The probability of monoclonality obviously also varies with the seeding density that's used in the experiment. So if we have an average number of cells per well of 0.1, the probability of monoclonality would be estimated to be about 95%. If we take a look at the plate, you can see that while the probability of monoclonality is high, there are a lot of empty wells on this plate. Again, basically consuming resources from your project. So if you use a higher seeding density, like an average of one cell per well, you have a lot fewer empty wells on the plate, but the probability of monoclonality is significantly lower, only about 58% for this case. And you can see that the average number, or the expected number of wells that contain one cell is significantly higher than with the lower seeding density. But you also have a lot more wells expected to contain more than one cell. And this is intuitive when you look at the plate. So although we have an average of one cell per well, there are still a lot of empty wells on the plate. And that's because a lot of the other wells are multiply seeded. Up until now, we've been looking at a homogeneous population of cells. But when you're doing limiting dilution cloning, you have two effective populations of cells. You have the cells that you are not going to select for further development, or uninteresting cells. And then those clones that are producing the product that you're interested in, that you're choosing to carry forward for further development, which we'll define as interesting cells. So it's really important to note that the distinction, the decision between these two is a binary decision, and it's made by the act of carrying forward a specific culture from a specific round of cloning. So you can use a variety of characteristics to decide whether or not you're going to carry a culture forward, obviously. So whether that be productivity of the culture, or affinity of the product, or specificity of the product, but once it meets that threshold that you choose to carry it forward for the next step in your cell line development process, that culture is defined as interesting for that round of cloning for the purposes of the statistics. So let's take a look at an example where we have a mixed population of cells where 30% of those cells are interesting. Obviously, this culture is also going to be distributed randomly over the plate, and this makes distributing the mixed population equivalent to the imaginary experiment of first distributing the uninteresting cells and then randomly distributing the interesting cells over the same wells. So that is to say that both the uninteresting and interesting cultures are distributed randomly over the plates. And furthermore, they're actually distributed with their own independent Poisson distributions. But because they're distributed over the same physical space, they exhibit overlapping Poisson distributions on the plates. And this means that some of the wells are going to be seeded with both interesting and uninteresting cells. And this was described by Robert Strudewski in the Yale Journal of Biology and Medicine. So what we're interested for obtaining our monoclonal culture that we'll use for downstream processes is the probability of a well containing one interesting cell and only one interesting cell. And this probability is equal to the probability of a well being seeded with one interesting cell multiplied by the probability of that same well containing 0 other cells. Both of these factors are equal to less than 1, and that means that the probability of monoclonality for an interesting culture will always be less than any random culture in the same experiment. This is the equation for the probability of a given well being seeded with a specific number of interesting and uninteresting cells. And that makes this the probability of monoclonality for an interesting culture. So using these equations on the right here and subbing them in and doing some algebra and moving around, we can arrive at this equation at the bottom here. And this is the equation for the probability of monoclonality for an interesting culture in terms of the uninteresting fraction, or the fraction of wells that we're not choosing to carry forward, and the fraction of empty wells. So again, you can take a look at that video tutorial if you want to see the derivation of these equations in more detail, but the take-home message for this section is that the probability of monoclonality for an interesting culture is always going to be less than the probability of monoclonality for any random culture. And to demonstrate this, let's take a look at an example. So in this case, you have an experiment where you're seeding 10 plates and observing a 30% cloning efficiency. And from this experiment, you're choosing 5 cultures that you're interested in carrying forward for further development. In this case, you'd be expected to have a probability of monoclonality for any random culture in the experiment of about 83%. However, for an interesting culture, for one of your 5 cultures that you're choosing to carry forward for further development, your probability of monoclonality is significantly less, only about 70%. And that means to achieve the probability of monoclonality that you'd be assuming if you're making the assumption that the probability of monoclonality is the same for any of the cultures, you'd actually have to either reduce your seeding density and thus, again, increase those wasted resources, or increase the number of cloning steps that you're doing during your cell line development process. So keeping that in mind, let's now switch gears back over to semi-solid cloning. So again, we have our independent cells that are individually suspended in the semi-solid medium and growing into those discrete colonies. So unlike limiting dilution cloning, where in order to obtain a monoclonal culture, the cells have to be placed in a specific spot, i.e. a unique well of the plate, no matter where those original cells are ending up on the culture dish in semi-solid cloning, they're going to grow into discrete monoclonal colonies. The exception to this, of course, is where those 2 original cells are placed so close together that when the colonies form, they actually overlap. The probability of monoclonality for semi-solid cloning is therefore related to the probability of overlap of the colonies on the plate. And this can be calculated by estimating the area of the plate that's covered by colonies divided by the total area of the plate. So I know I've been talking about a three-dimensional growth environment when it comes to semi-solid cloning, and suddenly I'm talking about area. But that is because when you're doing the picking, whether you're doing it manually or robotically, you're picking the colonies from above. So if they superimpose on the area of the plate, even if they're not physically touching in space, it's still going to be difficult to isolate those cultures independently. Therefore, we can talk about area when estimating the probability of monoclonality. So to calculate that probability of overlap, we need to know the area of the plate, the size of the colonies on the plate, and the number of colonies that are on the plate. And that's going to be able to allow us, rather, to estimate the probability of overlap for a colony. So this is the equation then that can be used to estimate the probability of monoclonality for a culture isolated in semi-solid cloning. So let's use this in an example. So let's take a 10-centimeter culture dish where we're growing 100 colonies, and the average colony diameter is about 0.75 millimeters. These colony diameters is typical of colonies that have been growing for about 10 to 14 days, depending on the cell type. So in this case, you'd have a probability of monoclonality of about 98% for a culture isolated in this experiment. And if you chose to do a second round of semi-solid cloning, that probability of monoclonality goes up to over 99.9%. Now, what's really important to remember when looking at these numbers is that they're derived from the equation that I showed on the previous slide, which assumes that all of the colonies on the plate are picked, regardless of their proximity to another colony or their shape or the proximity to the side of the dish. However, in practice, you can use criteria to exclude cultures that are less likely to be monoclonal, thus increasing the probability of monoclonality for cultures isolated in the experiment. So we can take a look at how you pick colonies in semi-solid cloning. So if you take a look at these two colonies here that are very, very close together, you can see how picking colonies that would be further spaced apart would give you a greater likelihood of those colonies that you're picking being monoclonal. So whether you're picking manually or using a robotic platform for your colony picking, you can choose to exclude those colonies that are less likely to be monoclonal, meaning that the estimates for the probability of monoclonality on the previous slide are actually underestimates for what you'd be expected to achieve in practice by excluding the colonies that are less likely to be monoclonal. And while in semi-solid cloning, you can use this judgment to increase the probability of monoclonality for your cultures, you can't do that in limiting dilution cloning, where selection of the cultures that you're choosing is a random process. So Clonacell is a stem cell technologies product line that includes a lot of semi-solid media for semi-solid cloning of various cell types. And at Stem Cell, we've developed a tool called the Clonacell Calculator that enables us to compare semi-solid cloning and limiting dilution cloning protocols in order to compare the probability of monoclonality and the resources used in order to isolate specific cultures. So I want to use this tool to compare a couple of different example protocols. So let's start with a limiting dilution cloning protocol where in your first round of cloning you seed 30 plates, observing a cloning efficiency of 20%. And from those cultures that grow, you're choosing 10 cultures to carry forward for subcloning. So then in subcloning, for each of those 10 cultures, you seed 196 well plate with a seeding density of 0.2 cells per well. And from this, choose 5 clones for further development. In limiting dilution, you'd be expecting a probability of monoclonality of about 96.4%, and that's cumulative over those 2 rounds of cloning. And in order to isolate those 5 clones, you're using 40 96-well plates. Now, if instead of using limiting dilution, you use semi-solid cloning to isolate the same 5 clones, you're going to see a probability of monoclonality closer to 97.8% in only 1 round of cloning. And instead of using 40 96-well plates, you are only going to require 6 to isolate those same 5 clones. And by same clones, I mean they're meeting the same criteria for productivity or affinity or whatever criteria you're using to select your clones. And I should mention actually that this tool is available on the Stem Cell Technologies website, and along with a link to the video tutorial, you'll be being sent a link to the clonocell calculator so you can input your own limiting dilution cloning protocols and compare what you would be expecting to see for using semi-solid cloning. So let's take just briefly a look at a second example that's on a slightly larger scale. So in this case, you're going to seed 100 plates in your initial round of cloning with a cloning efficiency of 15% after the cultures have grown. And from that first round, you're choosing 30 cultures to subclone. In both subcloning rounds, for each of the cultures that you're subcloning, you're seeding a single 96-well plate. And in the first subcloning round, using a seeding density of 0.2 cells per well, and choosing 10 cultures for your second subcloning round. And then using a seeding density of 0.1 cells per well to finally isolate, again, 5 clones. So for this protocol, you'd be expecting a probability of monoclonality of about 99.7%. And again, this is achieved through 3 cloning rounds using 140 96-well plates. If instead of limiting dilution, you went with semi-solid cloning, in only 2 rounds of cloning, you'd be expecting a probability of monoclonality of over 99.9%. And again, the numbers that are estimated using this calculator are an underestimate for semi-solid cloning of the probability of monoclonality you could expect to achieve in practice. So semi-solid cloning, we're seeing over 99.9% probability of monoclonality, 2 cloning rounds, and requiring only 20 96-well plates compared to 140 to isolate the same 5 cultures. So these results, or these estimations, demonstrate that there can be a lot of time and resources saved using semi-solid cloning instead of limiting dilution cloning during the cell line development process. And these saved resources can be used to either increase the scope of the projects that you're currently working on, or to choose to take on a greater number of projects in the same given time period. So I'll just give a brief summary of what I've talked about today. So for limiting dilution cloning, went through some of the reasons why the probability of monoclonality is lower for your clones of interest than for any random cultures in the same experiment. And that means that it's really easy to overestimate the probability of monoclonality for the cultures that you're most interested in your experiment. And semi-solid cloning makes it really easy to isolate cultures with a very high probability of monoclonality. And this is increased even further because the actual procedure of picking the colonies can be biased towards selection of cultures with an even higher probability of monoclonality. And because you have a high probability of monoclonality for each round, you require fewer rounds of cloning and therefore shorter cell line development timelines. We also touched on how semi-solid cloning is able to isolate pools of cultures with very high diversity and growth characteristics, making it easy to find those high-producing clones, and that fewer physical resources are needed for semi-solid cloning with respect to, in comparison to limiting dilution cloning, allowing you to reallocate those resources elsewhere.